Bismillahir Rahmanir Rahim. Good morning, class. This is our third lecture on Chapter 2. Chapter 2 is about the science of the cell, which we defined as cytology. Last time we said that cytology is a term that made of two words, cyto, which means cell, and ology, which means science. Also, in the last lecture, we denied that by the term cell, we do not mean the cell phone nor the detained cell in jail. We define cell as the smallest structural and functional unit of the organism. It is to say, of the life. Also, we agreed that organisms are either unicellular organism, that means the whole body of the organism is made of one cell, or a multicellular organism, that means the organism is made of more than one cell, might be millions. We have two examples of this. We say that bacteria is an example of a unicellular organism, and mammals as well as birds are examples of multicellular organisms. Also, we agreed that our backbone course book is going to be the biology of Cambridge IGCSE, and the fourth edition. Why the fourth edition? Because it is the newest edition, and in the meanwhile, this edition is exactly as the curriculum of the years 2023 through 25. So by this, we put the framework of how we are going to move forward, or how we will move forward towards this course. Also, I would like to notice that this course is going to be more appropriate for the students who are going to take their biology exam next year. I mean on May, June 23, and on. Why? Because there is a change on this syllabus, and this will not be appropriate for the students who will take their exam next October or November. But anyhow, the earlier, or the maybe the five foremost chapters may be of help to them. Let us move forward and see what's going on. In our previous lecture, we emphasized that what in that lecture is all features that appears to you when you examine a specimen under light microscope. And under light microscope, because there is a limit of magnification, you cannot see all details. Biologists do not stop there. They go forward. The biology engineers invented what is called the electron microscope. Electron microscope is little bit actually far different from the light microscope. The light microscope uses the light to visualize the sunlight or the lamp light in order to visualize the objects. But the electron microscope had different techniques. It used electron to magnify the objects to more than far more than the light microscope. We are gonna study this in details when we come to microscopy and how we can visualize things that you cannot see by your naked eye. And by electron microscope and the big magnification, biologists or cytologists can see more and can examine more, can see more details and identify new organelles inside. And this is why I would like you to be sure when the questions come in the test. Do they ask you about light microscope, the feature to be determined? Are they in a light microscope or in an electron microscope? Because they differ. In electron microscope, we have more spectrum to see, more details to see. And for example, this is a sketch of animal cell under electron microscope. Also, electron microscope is very huge and big. It's like this one. You can see it here. It's not like the normal microscope. I mean the size and also the technique. Let us go forward. Here we are going to study information that are present in your book and also under electron microscope. In your course book, there are two organelles that can be seen if we visualize them under electron microscope. The first one is the mitochondrion, and a mitochondrion is a singular word. The plural word is mitochondria. So when you hear a mitochondria, that means there is more than one mitochondrion. Those are the small organisms or organelles, and last time we defined organelles as a small organ with a double membrane arrangement. The outer membrane, the intermediate space, the inner membrane are called cristae, and also cristae is the plural, crista is the singular, are folded, and when something folds, it increases the surface area for electron transport. And by means of electron transport, this is going to be happened, and we are going to talk about this in details when we come to respiration. Found in all cells except prokaryotes, so bacteria is deprived of mitochondria. All prokaryotes are deprived of mitochondria. More abundant in cells that use more energy. Why? Because actually when we come to the function, their main function is to produce energy. So they are abundant, they are many, they are much in number, they are huge in number compared to other cells. When this cell is a cell that needs energy, so it is abundant in muscle cells, in sperm cells, and also nerve cells. Why? Muscle cells, it is needed. Those mitochondria are needed in abundance or in much number, more number, because they are contracting every time. Our movement depends on them. The sperm cell needs more mitochondria, because that means they need more energy, because they have to swim inside the female genital tract in order to make proper fertilization. Nerve cells also need that, because nerve cells all the time they are triggered and they have to move information from the brain to the organs, and this needs more energy. We are now explaining why they are needed in abundance in such cells. The second organelle that present in your book is the ribosome. Ribosomes are the plural. The ribosome is a tiny organelle. In size, it is less than smaller than the mitochondrium, with no membrane and made from RNA and proteins. Actually, with one membrane. It consists of two separate RNA protein complex known as the small and the large subunits. And if you see here, it looks like this. You see, it looks like here, it looks like you put your two hands like this. One is the smallest, the bottom is the smallest unit, subunit, and the above or the big one or the upper one is the large one, with an RNA template sandwich between them. Sandwich means between them, like your burger sandwich. You see, you have two bread together and there is a sandwich inside them, the burger itself. Some of the ribosomes are bound to ER. ER is an abbreviation of endoplasmic reticulum. Endoplasmic reticulum. And the others are free. So I have two types of ribosomes. One of them are tied or bound to endoplasmic reticulum. We are going to talk about it later. And the others are free. Free means that they swim inside the cytoplasm freely without being tied to any other organelles. One more thing I would like you to notice is that if you see this mark, that means this information present in your book. It is part of your course book. If you see the stop sign, we are going to see it after few minutes. That means this information is an added information, and I add them because I believe that they are important for your knowledge and for your understanding. They are not in your course book, but you have to understand them as far as my knowledge concerned, and you have the you are free if you want to take them or leave them. Let us move forward. In the last scene, we studied the structure of the two organisms that are present in your book, the mitochondria and the ribosomes. And now we would like to study the function of these two organelles. The most important thing I would like you, every time you remember that the structure matches the function. It is very, very important. You have to tie them together. Whenever you have a special structure, that means you have a special function. Okay, so mitochondrion is this site of aerobic respiration. By aerobic respiration, we mean that the respiration is an oxygen dependent respiration. It requires oxygen in order to be fulfilled. Without oxygen, this is not aerobic respiration, it is anaerobic respiration. It is something else. We are going to study it sometime. Okay, so it is the site of aerobic respiration. Very important to understand this. Produces ATP, and ATP stands for adenosine triphosphate. Adenosine triphosphate. Adenosine triphosphate. It is release energy. Oxygen is used to release energy from glucose. Glucose is produced from starch in plant cells and from glycogen in animal cells. There is the big why. Why? Because the storage carbohydrate in plant is starch, while the storage carbohydrates in animal is glycogen. So animal cannot use starch because starch is not stored inside the animal body as starch, and also plants cannot use glycogen because plants do not store glycogen as glycogen. Maternally inherited. This is an important character because mitochondria comes from the maternal portion of the person, of the organism, from the mother, not from the father. Maternal means from mother, not from the father. Then the ribosomes. The second one is the ribosomes. Ribosome is important for protein synthesis, so it is responsible for protein synthesis. The second thing is important, the second character, it is found in abundance in cells that secrete secretions of protein nature, in cells that produce enzyme hormones, etc., that are chemically proteins. What do I mean by that? If ribosome produces protein, and I have a special cell that produce, for example, an enzyme or hormone, and the enzyme or hormone are chemically, when we go and see chemically from where what they are constituent from, or their constituents are what they are. Chemical nature of these constituents are protein. So if this is the case, that means the special cell produces a special type of protein. We are going to see more ribosomes inside this cell. Why I'm saying that? Because you are going to be asked about that. And also you are going to be asked about the abundance of mitochondria. Be careful about those two, because those two organelles are essential for your curriculum. They are present in your course book, and you will be asked about at least one of them if they are not the two. However, when you see, again, when you notice that this sign, the check sign, green check sign, that means this information is provided by your book and you have to know it. You have no choice. You have to know it. Let us go forward. Before we proceed, I would like you to see and notice the stop sign. When you see this stop sign in any slide or information, that means this information is an added information. The information by itself is not found in your course book. But I do believe that it is important for your knowledge. You are free to take it or leave it as you like, but I would like you to understand that it is important and you have to know it in order to be able to manipulate information and tie them together and solve the puzzle. Still, we are talking about the under electron microscope appearance of the object or the cell here. Our object here is the cell. So we are going to take what we call it endoplasmic reticulum. Reticulum means net. When you hear the word reticulum means net. So the endoplasmic reticulum is, endo means inside, plasmic means in the cytoplasm. So this is the net that present inside within the cytoplasm. This net has two appearances, either a rough endoplasmic reticulum, and it is abbreviated, or the acronym of it is RER, rough endoplasmic reticulum, or a smooth endoplasmic reticulum, which is SER. Why they name it rough and smooth? Because the rough endoplasmic reticulum contains ribosomes attached to its surface, making the rough appearance. It is adjacent to the nucleus and found in all eukaryotes, and it is a flattened membranous sac rose. The smooth endoplasmic reticulum is a little bit different. It is the same, it's flattened membranous sac rose, but there is no ribosomes inside it. And because there is the difference, the main difference is in the presence or absence of ribosomes attached to its surfaces, there are differences in their function. We are going to see them in the next slide. In the previous slide, we studied the structure of the two endoplasmic reticulums. Here, we are going to study their function. And when we talk about the function, we have to remember that the structure matches the function. So the endoplasmic reticulum is important for protein synthesis and also for membrane synthesis. And this means folding, modification, and transport of proteins, which is a very important. And this is why I believe that it is important to know what an endoplasmic reticulum is. The second one, the smooth endoplasmic reticulum, it has different function. Why? Because it is deprived of ribosomes. So the first one is found, the first function is the detoxification of drugs in the liver cells. So the liver cells are in abundance in this liver, and also it has a voluminous type of SER, which is a smooth endoplasmic reticulum. Why? Because it used them for detoxification. Detoxification, that means render drugs non-toxic. Okay, change the drug from toxic drugs into non-toxic drugs. Or from toxic compound to non-toxic compounds. Again, it is responsible for the synthesis of lipids, including cholesterol and phospholipids, which are used in the production of a new cellular membrane. And this is important. Plays an important role in the synthesis of steroid hormones. Why steroid hormones? Because it is found from the cholesterol, and it is important to form cholesterol itself. So it is important, as in an adrenal glands. Also, it regulates calcium ion concentration on the cytoplasm of striated muscle cells. It is very important to know that things. Let us move forward. Another two organelles are also important. Again, when you see the stop sign, you have to know or notice that this information is an added information. It is not present in your course book curriculum, so be careful about that. Golgi apparatus or Golgi body or Golgi complex or Golgi complex are flattened membranous sacs arranged in rows. This is from the structure point of view. Lysosomes. Lysosome is a membrane-bounded spherical or sac-like organelle, contains digestive enzyme. We are going to see their main function in the coming Golgi apparatus and the lysosomes functions are important. Golgi apparatus modifies and transport proteins, synthesis of carbohydrates. So they are important to functions. Lysosomes, phagocytosis and intracellular digestion, the degradation and recycling of cellular waste, and digestion of intracellular material. And this is why they are in abundance. You found them in abundance in the neutrophil cells. The neutrophil cells are cells that are important to map all the bad guys present in the body, and they are an innate source of immunity in the body. And phagocytosis is the main picture of them, the main function of them. Autophagy or apoptosis or self-eating. The self itself needs to be generated, so sometimes there is a self-eating of the cells itself, and it's called autophagy or autophagy, and also it's called apoptosis. This is, and now you can understand why I add those information. Stop sign is there. Now we come to the end of this lecture. Thank you for having me, and thank you for listening. I appreciate your patience, and hope to see you next time. Have a good day, and bye-bye.